Okay, students, so this is course CAM 4451 and this is lecture 6. The topic is antibacterial drugs and mechanism of action part 2, and I am Dr. Musab Ibrahim, Assistant Professor, Department of Chemistry, CAST. So now the topics to be discussed in this lecture include mode of action of antimicrobials, that is related to the mechanism of action of antibiotics working on different targets of the bacteria. Protein synthesis inhibitors. So those antimicrobials are those antibiotics which attack the process of protein synthesis in bacteria. DNA replication inhibitors. So those antibiotics are those antimicrobials which attack the DNA replication process of the bacteria and as a result stop the DNA replication process in bacteria. RNA synthesis inhibitors. So those antimicrobials or antibiotics which stop the RNA synthesis in bacteria. Metabolic pathways inhibitors. So those antibiotics or antimicrobials which attack different metabolic pathways in bacteria and as a result stop those metabolic pathways. And cell wall integrity inhibitors. So those antimicrobials or antibiotics which attack the cell wall integrity and as a result damage or disintegrate the cell wall of the bacteria. So the mechanism of action, one of the mechanism by which the antibiotics or drugs acting on bacteria is by inhibiting the protein synthesis. This is by inhibition of protein synthesis. So the ribosomes of the bacteria are the target of the antibiotics which the antibiotics or antimicrobials bind, and as a result the ribosomes they cannot perform their proper function, and so the protein synthesis process is stopped. So does the growth and production of the bacteria also stops. And the drug of this class include aminoglycosides, macrolides, chloramphenicol, lincosamides, tetracyclines, oxazolidinones, streptogramines. So let's have a look at the mechanism of protein synthesis and how does the antibacterials or antibiotics they are disturbing or stopping the process of protein synthesis. So here we can see that the first step is the DNA in nucleus serves as a template, and then in the second step the messenger RNA is processed before leaving the nucleus. So here you can see that the DNA template is working as a template, and then RNA is being synthesized, and then after processing the RNA is being processed and then sent outside the nucleus. So in the third step, when messenger RNA is formed, it has codons, and then in the fourth step you can see that the messenger RNA moves into cytoplasm and becomes associated with ribosomes. So this is the messenger RNA, and this is the ribosomal subunit 50S, and this is the ribosomal subunit 30s. So here you can see that in the fifth step, the transfer RNA, which has anticodons and it has attached specific amino acids, so that carries amino acids to the messenger RNA. So these are the anticodons, and this is transfer RNA, and this is the amino acid which is attached to the transfer RNA, and this is the pool of amino acid which is present within the cytoplasm. So then in step 6, what is happening is anticodon-codon complementarity base pairing is taking place. So here we have the codon there, and then we have the anticodon on the transfer RNA. So on the messenger RNA, the codon is present, and on the transfer RNA, the anticodon is present along with the amino acid. So what is happening is next step, which is step number 7. So peptide chain is transferred from resident transfer RNA to incoming transfer RNA. So what is happening is that this peptide chain, which has already been in the process of synthesizing, so that is being transferred from the already present transfer RNA, which is this one, and then this peptide chain is transferred to this amino acid, and then in the 8th step, the transfer RNA departs and will soon pick up another amino acid. So again, this transfer RNA goes to the amino acid pool, and then it attaches again a complementary amino acid, and then again it comes to the ribosomal subunit, and this is how this cycle is repeated, and as a result the elongation of the peptide chain is taking place. So this is actually the process of protein synthesis by way of messenger RNA and transfer RNA, and the purpose of letting you know and describing this slide is to understand that there are different areas, different steps. So if you look at step number 1, then step number 2, which is mRNA processing, then step number 3, where you have messenger RNA has codons, then step number 4, in which messenger RNA is associated with the ribosomes. So starting from step number 1 up to step number 8, these are the different sites at which the antimicrobials are acting, and as a result of their action, either at step number 1 or 2 or 3 or 4 or 5 or 6 or 7 or 8, so this results in stopping the process of protein synthesis in bacteria, and that is the mechanism of action of different drugs of choice, which we will be discussing in the coming slides. So here you can see that there are different antimicrobials which are responsible for stopping the protein synthesis process in bacteria. So if you look at the aminoglycosides, so what they are doing is they are blocking the initiation of translation, and causing the misreading of messenger RNA. So the aminoglycosides, they attach themselves to the 30S ribosomal subunit of the ribosome, the bacteria, and they block the initiation process of protein synthesis, and as a result cause the misreading of messenger RNA. So if you look at the tetracyclines, so they block the attachment of the transfer RNA to the ribosome. So again, they are attaching themselves to the 30S ribosomal subunit of the ribosome of bacteria, and that's how they stop the process of protein synthesis. Now moving towards streptogramins. So these streptogramins, they each interfere with a distinct step of protein synthesis, which we will be studying and discussing in the coming slides. So the macrolides, they prevent the continuation of protein synthesis, which means that the peptide bond formation is being prevented by the macrolides, and they attach themselves to the 50S ribosomal subunit of the ribosome of bacteria. Moving towards chloramphenicol, so it also prevents peptide bonds from being formed. So as we know that the formation of peptide bonds is very important in the protein synthesis. So if that is stopped, so then there is no protein synthesis taking place. And again, the chloramphenicol, their mode of action occurs at binding to the 50S ribosomal subunit. Now lincosamides, so they prevent again the continuation of protein synthesis and also bind to the 50S ribosomal subunit. And then is the oxos already known, and they interfere with the initiation of protein synthesis, which is the starting process of protein synthesis. Okay, so the mechanism of action, protein synthesis inhibitors, and now about the aminoglycosides. So what the aminoglycosides do is that they irreversibly bind to the 30S ribosomal subunit, and as a result of binding to the 30S ribosomal subunit of the ribosome of bacteria, it blocks the initiation, translation, and causes misreading of messenger RNA. And this is the mechanism, and these aminoglycosides are not effective against anaerobes, enterococci, and streptococci species, and often they are used in synergistic combination with beta-lactam drugs. So synergistic combination means that when drugs are used in combination, two or more than two antibiotics are used at the same time so that the disease or infection can be cured. The examples of aminoglycosides includes gentamicin, streptomycin, and tobramycin. So if the drug is used for prolonged periods, so then the side effects can include nephrotoxicity and ototoxicity. So nephrotoxicity is the toxic effects on the kidney, and ototoxicity is the toxic effects on the ear. So here you can see that these are the aminoglycosides, and this is the attachment site at 30S ribosomal subunit of the bacteria, and this is the messenger RNA. So as a result of attachment of these aminoglycosides, at this very specific site of the 30S ribosomal subunit. So the initiation process and the translation process both are being stopped, and as a result the mRNA is misread and the protein synthesis which is taking place is not correct. So the correct protein synthesis is not taking place. Right, so the next one is tetracycline, and the mechanism of action of tetracycline is that they reversibly bind to the 30S ribosomal subunit of the ribosome of bacteria, and as a result of this binding, it blocks the attachment of transfer RNA to the ribosome. And when the attachment of transfer RNA to the ribosome is not taking place, so as a result, the protein synthesis continuation is being stopped. So these tetracyclines, these are narrow range and effective against certain gram-positive and gram-negative bacteria. So here you can see that these are the tetracycline, sorry, and this is the site of attachment where they are attaching themselves to the 30S ribosomal subunit, and this is the messenger RNA, and as a result of this attachment of the tetracycline, the tRNA is blocked. So the transfer RNA, the incoming transfer RNA, it cannot attach itself, and as a result the further process of protein synthesis continuation can no longer be happening, and as a result the protein synthesis process is stopped. Right, so moving towards macrolides, so these antibiotics, they reversibly bind to the 50S ribosomal subunit of the ribosome of bacteria, and as a result of their binding they prevent the continuation of protein synthesis. These macrolides are effective against a variety of gram-positive organisms, and these are often drug of choice for patients which are allergic to penicillin. So those patients who cannot use penicillin, so then they are being given macrolides. And the examples include erythromycin, clarithromycin, and azithromycin. So the resistance against these drugs can be developed by the bacteria through the modification of RNA target. So the target at which the antibiotic binds, so if that target is modified, then the drug can no more bind to the target. As a result, the resistance is developed against that specific drug by the bacteria. So here you can see that this is the examples of erythromycin. So it is binding at this site to the 50S ribosomal subunit of the ribosome of bacteria, and as a result the ribosome is prevented from translocating, and this is the messenger RNA. So what is happening is that when the erythromycin has bound at this target site, so then this transfer RNA, it is not being able to get detached, and as a result the incoming transfer RNA is also not able to go to the next stage, and as a result the protein synthesis process is stopped. Right, so moving towards chloramphenicol, which is another antimicrobial drug whose mode of action is also by stopping the protein synthesis process. So The mechanism is by binding to the 50S ribosomal subunit of the ribosome of bacteria, and as a result preventing the peptide bond formation. And this chloramphenicol is wide spectrum antibiotic, which means that it is effective against a wide range of gram-positive and gram-negative bacteria. This is also considered to be a drug of last resort because when all the other drugs they are not working, so then the chloramphenicol is administered to the patient. And sometimes a very dangerous side effect has been observed in patients who are using chloramphenicol, which is aplastic anemia, which is related to the deficiency and damaging of the red blood cells. So here you can see that this is the chloramphenicol, and it is attaching itself at this site and preventing the peptide bond formation between two amino acids, and as a result the protein synthesis process is stopped. Right, so now we are discussing the lincosamides. The example is clindamycin, and the mechanism of action of lincosamides is by binding to the 50S ribosomal subunit and thereby preventing the continuation of protein synthesis. So this lincosamide, it inhibits a variety of gram-positive and gram-negative bacteria, and useful in treating infections from intestinal perforation. So the lincosamides, they are specifically used in intestinal infections when there is perforation taking place inside the intestines, and it is very effective against bacteroides fragilis and Clostridium difficile. Right, so there is a new class effective against beta-lactams and vancomycin resistant gram-positive forms. One of them is oxazolidinones, and the mechanism of action of oxazolidinones is by binding to the 50S ribosomal subunit and thereby interfering with the process of protein initiation. The another one is streptogramins, and the mechanism of action of streptogramins is binding at two different sites on 50S ribosomal subunit, and thus by stopping the protein synthesis process. Right, now moving towards mechanisms of action. So some of the drugs, they stop the DNA replication process in bacteria, and thereby they stop the growth of the bacteria. So fluoroquinolones are the examples which stop the DNA replication process, and the process is inhibiting the action of topoisomerase DNA gyrase. So that is the enzyme which maintains the supercoiling of DNA, and as these fluoroquinolones, so they are responsible for the process of supercoiling, and as a result of fluoroquinolones the supercoiling of DNA is not happening, and as a result the DNA replication is also not taking place. And these fluoroquinolones are broad spectrum antibiotics effective against a large variety of gram positive bacteria as well as gram negative bacteria. And the examples include ciprofloxacin, which is a very common antibiotic, as well as the ofloxacin. Sometimes the resistance is developed against these fluoroquinolones antibiotics due to the alteration of DNA gyrase. So the DNA gyrase, when there is alteration in the structure of the DNA gyrase, so then the fluoroquinolones, they can no longer bind to the DNA gyrase, and as a result the medicine or the antibiotic, it cannot work on the process of preventing the supercoiling of DNA, and as a result the medicine or the antibiotic is no more effective. So that is how the resistance is developed by certain bacteria against this antibiotic, which are fluoroquinolones. Right, so rifamycins are a class of antibiotics which stop the RNA synthesis of the bacteria, and their mechanism of action includes by blocking the RNA polymerase of the prokaryotic cells, so basically of the bacteria. So the RNA polymerase is blocked, and this RNA polymerase is responsible for the initiation of transcription, which is actually the conversion of DNA into RNA. So that is the mechanism of action of rifamycins, and it is most widely used among rifamycins is rifampin, and it's broad spectrum, effective against a variety of gram-positive bacteria as well as gram-negative bacteria, and some of the mycobacterium species as well. And the rifamycins are used for treating tuberculosis, meningitis, and Hansen disease. So some of the drugs, they inhibit certain metabolic pathways of the bacteria and thereby stopping the growth of the bacteria. So one of them is folate inhibitors, and the mode of action is to inhibit the production of folic acid. So that is how they are stopping the growth of the bacteria. So they mimic the PABA, which is stands for para-aminobenzoic acid, and the antimicrobials in this class include sulfonamides, trimethoprim, and human cells lack specific enzymes in folic acid pathway. Therefore, when the patient is taking that drug, so he is not being affected by this drug, because this drug is only effective on the enzymes which are present in the metabolic pathways of the bacteria. So here you can see that this is the structure of the para-aminobenzoic acid, and this is the structure of the sulfanilamide, which is a sulfur drug. So the only difference between para-aminobenzoic acid and sulfanilamide is this functional group, which is SO2NH2. So here you can see that in the metabolic pathway, the precursor 1 is converted into precursor 2, and so and this process is catalyzed by enzyme. So here the sulfur drugs, they are acting as inhibitors and stopping the process of this first step in the metabolic pathway, and from precursor 2 you are getting dry hydrofolate, and that is being catalyzed by enzyme number 2, and then from dihydrofolate you are getting tetrahydrofolate, and this is being catalyzed by enzyme number 3, and trimethoprim is the inhibitor or the drug which stops this reaction, and as a result there is no tetrahydrofolate synthesis taking place, and as a result of further metabolic pathways These tetrahydrofolate are responsible for thymine, guanine, adenine nucleotides. So when the sulfur drugs at step number 1 and trimethoprim at step number 3, so when they stop this metabolic pathway, so as a result the nucleotide synthesis is stopped, and as a result the bacteria cannot reproduce, they cannot grow, and that's how the drug is acting on the bacteria. Right, so some of the antibiotics, they are involved in damaging the cell wall integrity of the bacteria. So polymyxin is B is the most common one, and it is common ingredient in first aid skin ointments, and binds to the membrane of the gram-negative cells of the bacteria. That is the specificity of this drug, that only binds to the gram-negative bacteria, and alters permeability, so it allows and expels certain uh, molecules or certain functional groups across the membrane, so that permeability is being altered by the application of polymyxin B, and also binds to eukaryotic cells. So this is the ability of polymyxin B, that it can bind to the human cells as well, and that is the why it is only being used as a topical application, so only on the skin, on the outer surface of the skin, but it is not being used to orally or in the form of injection. So this is the process. So this is polymyxin B, and this is the cell wall of the bacteria, and this is the cell membrane. So when the polymyxin is applied, so it goes across the cell wall and then enters the cell membrane. So here you can see that it is passing through the cell membrane, and as a result the cell membrane integrity has been compromised, and there are holes, and as a result the cell membrane is no more in its original form, and as a result the bacteria, it cannot maintain its process of permeability of other molecules to pass through it, and eventually the bacteria, it gets killed. So these are the references, and for further reading and explanation you can consult these books, and thank you.